I fully admit that I lack the philosophical knowledge to dispute the moral framework of the vegan-vegetarian debate. However, I would like to present a series of studies showing that these diets do in fact have health risks that are unique and potentially dangerous. I do not deny the existence of health risks associated with omnivorous diets, nor have I ever. This is not the be-all, end-all of arguments against alternative diets, but the following negative effects pose a problem. Ultimately, the diet of each individual person will vary, and with proper supplementation and doctor supervision, it is possible to sustain life. It should be noted that an increase in health consciousness amongst all dietary regimes, whether extreme as veganism or vegetarian or not, will increase life longevity and overall fitness. I'll start out with talking about primary nutritional deficiency. For example, vitamin B12 is a water-soluble vitamin that is involved in the function of every cell in the body. It is particularly important in the formation of blood and the function of the brain. Even with supplementation, this study has shown 92% of vegans are still deficient of this critical nutrient. Please refer to source number 1. Proteins. Let's talk proteins and the Hoffman-Falvo study on protein sources. Proteins are nitrogen-containing substances that are formed by amino acids. They serve as the major structural component of muscle and other tissues in the body. In addition, they are used to produce hormones, enzymes, and hemoglobin. Proteins can also be used as energy. However, they are not the primary choice as an energy source. For proteins to be used by the body, they need to be metabolized into their simplest form, amino acids. There have been 20 amino acids identified that are needed for human growth and metabolism. 12 of these amino acids, 11 in children, are termed non-essential, meaning that they can be synthesized by our body and do not need to be consumed in the diet. The remaining amino acids cannot be synthesized in the body and are described as essential, meaning that they do need to be consumed in our diets. The absence of any of these amino acids will compromise the ability of tissue to grow, be repaired, or be maintained. The composition of various proteins may be so unique that their influence on physiological function in the human body could be quite different. The quality of a protein is vital when considering the nutritional benefits that it can provide. Determining the quality of a protein is determined by assessing its essential amino acid composition, digestibility, and bioavailability of amino acids. This is according to the World Health Organization 1990. There are several measurement scales and techniques that are used to evaluate the quality of protein. Numerous methods exist to determine protein quality. These methods have been identified as protein efficiency ratio, biological value, net protein utilization, and protein digestibility corrected amino acid score, or PDCAAS. Protein is available in a variety of dietary sources. These include foods of animal and plant origins, as well as the highly marketed sports supplement industry. Determining the effectiveness of a protein is accomplished by determining its quality and digestibility. Quality refers to the availability of amino acids that it supplies, and digestibility considers how the protein is best utilized. Proteins from animal sources, i.e. eggs, milk, meat, fish, and poultry, provide the highest quality rating of food sources. This is primarily due to the completeness of proteins from these sources. Although protein from these sources are also associated with high intakes of saturated fats and cholesterol, there have been a number of studies that have demonstrated positive benefits of animal proteins in various population groups. Campbell, 1999, Godfrey, 1996, Panemons, 1998. Protein from animal sources during late pregnancy is believed to have an important role in infants born with normal body weights. Godfrey, 1996, examined the nutrition behavior of more than 500 pregnant women to determine the effect of nutritional intake on placental and fetal growth. They reported that a low intake of protein from dairy and meat sources during late pregnancy was associated with low birth weights. In addition to the benefits from total protein consumption, elderly subjects have also benefited from consuming animal sources of protein. Diets consisting of meat resulted in greater gains in lean body mass compared to subjects on a lacto-ovo-vegetarian diet. Campbell, 1999. High animal protein diets have also been shown to cause a significantly greater net protein synthesis than a high vegetable protein diet. Panemons, 1998. This was suggested to be a function of reduced protein breakdown occurring during the high animal protein diet. However, we cannot deny there is a number of health concerns raised concerning the risks associated with protein emanating primarily from animal sources. Primarily, these health risks have focused on cardiovascular disease. Whey is a general term that typically denotes the translucent liquid part of milk that requires following process, coagulation and curd removal, of cheese manufacturing. From this liquid, whey proteins are separated and purified using various techniques yielding different concentrations of whey proteins. Whey is one of the two major protein groups of bovine milk, accounting for 20% of the milk, while casein accounts for the remainder. All of the constituents of whey protein provide high levels of essential and branched-chain amino acids. The bioactivities of these proteins possess many beneficial properties as well. Additionally, whey is also rich in vitamins and minerals. Whey protein is most recognized for its applicability in sports nutrition. Additionally, whey products are also evident in baked goods, salad dressings, emulsifiers, infant formulas, and medical nutritional formulas. Whey is a complete protein whose biologically active components provide additional benefits to enhance human function. Whey protein contains an ample supply of the amino acid cysteine. Cysteine appears to enhance glutathione levels, which has been shown to have strong antioxidant properties that can assist the body in combating various diseases. Kunus, 2000. In addition, whey protein contains a number of other proteins that positively affect immune function as well as antimicrobial activity. Ha and Zemmel, 2003. Whey protein also contains a high concentration of branched-chain amino acids, or BCAA, that are important for their role in the maintenance of tissue and prevention of catabolic actions during exercise. This is from McLean, 1994. Casein is the major component of protein found in bovine milk, accounting for nearly 70 to 80% of its total protein and is responsible for the white color of milk. It is the most commonly used milk protein in the industry today. Milk proteins are of significant physiological importance to the body for functions relating to the uptake of nutrients and vitamins, and they are a source of biologically active peptides. Similar to whey, casein is a complete protein and also contains the minerals, calcium, and phosphorus. Casein has a PDCAAS rating of 1.23, which is generally reported as a truncated value of 1.0. This comes from Dutz, 1998. Conclusions from this study. I will also recommend reading on the soy part of the study, which comes after the casein part. But our conclusions from this study. It does appear that protein from animal sources is an important source of proteins for human from infancy until mature adulthood. However, the potential health concerns associated with a diet of protein consumed primarily from animal sources should be acknowledged. With a proper combination of sources, vegetable proteins may provide similar benefits as protein from animal sources. Maintenance of lean body mass, though, may become a concern. All of this can be found in source number 2. Let's look at the dietary animal protein intake study in older women. Aging is associated with reductions in muscle mass and strength, so-called sarcopenia, and is generally characterized using muscle mass index, or MMI. It is believed that adequate nutrition, especially regarding protein intake, can delay this progression and enhance the quality of life of elders. We examined whether the predominant source of protein consumed, animal versus vegetal, by older women was associated with MMI. Here's our results. Significant correlations were observed between MMI and body mass index, fat-free mass, muscle protein content, total protein intake, animal protein intake, fat mass, visceral fat, and daily energy expenditure. However, a stepwise regression analysis showed an animal protein intake to be the only independent predictor of MMI. Our conclusions. These results suggest that protein intake, especially from animal sources, may be associated with a better preservation of MMI. However, more research is needed to confirm our results. This is from source number 3. Effects of an omnivorous diet versus an LVO or vegan diet in older men. The objective of this study was to assess whether the consumption of an omnivorous meat-containing diet would influence resistance training-induced changes in whole body composition and skeletal muscle size in older men compared with LOV vegan meat-free diets. Results. Maximal strength of the upper and lower body muscle groups that were exercised during resistance training increased by 10 to 38% independent of diet. The RT-induced changes in whole body composition and skeletal muscle size differed significantly between the mixed and LOV diet groups. With resistance training, whole body density, fat-free mass, and whole body muscle mass increased in the mixed diet group, but decreased in the LOV diet group. Type 2 muscle fiber area of the vastus lateralis muscle increased with resistance training for all men combined, and the increase tended to be greater in the mixed diet group than in the LOV diet group. Our conclusion. Consumption of a meat-containing diet contributed to greater gains in fat-free mass and skeletal muscle mass with resistance training in older men than did an LOV diet. This is from source number 4. Let's look at the relationship between animal protein intake and MMI in healthy women. The amount and the type of dietary protein could play a role in determining the quantity of skeletal muscle mass. The aim was to examine the relationship between the type of protein intake and the level of muscle mass in healthy omnivorous and vegetarian Caucasian women. The design of the present study was an observational and cross-sectional study. Animal protein intake was the independent predictor of muscle mass index. Thus, a vegetarian diet is associated with a lower muscle mass index than is an omnivorous diet at the same protein intake. A good indicator of muscle mass index in women seems to be animal protein intake. This is from source number 5. Creatine and muscle performance in vegetarians. The purpose of this study was to compare the change in muscle creatine, fiber morphology, body composition, hydration status, and exercise performance between vegetarians and non-vegetarians with 8 weeks of creatine supplementation and resistance training. Our results. Biopsy samples indicated that total creatine was significantly lower in vegetarians than non at the baseline. The change in muscle total creatine was significantly correlated with initial muscle total creatine and the change in lean tissue mass and exercise performance. These findings confirm an ergogenic effect of creatine during resistance training and suggest that subjects with initially low levels of intramuscular creatine, in this case vegetarians, are more responsive to supplementation. This comes from source number 6. The importance of carnosine as an anti-aging drug in humans. This is a 2-part. Carnosine is an endogenous free radical scavenger. The latest research has indicated that apart from the function of protecting cells from oxidation-induced stress damage, carnosine appears to be able to extend the lifespan of cultured cells, rejuvenate senescent cells, inhibit the toxic effects of amyloid peptide, or A-beta, malone dialdehyde and hypochlorite to cells, inhibit glycosylation of proteins and protein DNA and protein-to-protein cross-linking, and maintain cellular homeostasis. Also, carnosine seems to delay the impairment of eyesight with aging, effectively preventing and treating senile cataract and other age-related diseases. Therefore, carnosine may be applied to human beings as a drug against aging. This is from source number 7. In relation to this, vegetarianism and reduced muscle carnosine levels in humans. Carnosine is found in high concentrations in skeletal muscles, where it is involved in several physiological functions. The muscle carnosine content measured within a population can vary by a factor of 4. The aim of this study was to further characterize suggested determinants of the muscle carnosine content, diet, gender, or age, and to identify new determinants, plasma carnosinase activity and testosterone. Vegetarians have a lower carnosine content of 26% in gastrocnemius, compared to omnivores. In contrast, there is no difference in muscle carnosine content between omnivores with a high or low ingestion of beta-alanine. Muscle carnosine levels are not related to the polymorphism of the CNDP1 gene or to the enzymatic activity of the plasma carnosinase. In conclusion, neither CNDP1 genotype nor the normal variation in circulating testosterone levels affects the muscular carnosine content, whereas vegetarianism, female gender, and an increasing age are the factors associated with reduced muscle carnosine stores. This is from source 8. Omega-3 deficiency in Austrian vegans. The unbalanced N6 and N3 ratio, this is omega-6 and omega-3 fatty acids, and the limited dietary sources of eicosapentaenoic acid, EPA, and DHA in vegans and vegetarians led to reductions in C20-5, N3, C22-5 N3, C22-6 N3, and total N3 fatty acids in SPL, PC, PS, and PE compared with omnivores and semi-omnivores. The total content of polyunsaturated fatty acids, monounsaturated fatty acids, and saturated fatty acids remained unchanged. Our conclusion: the vegetarian diet, with an average N6-N3 ratio of 10:1, promotes biochemical N3 tissue decline. To ensure physical, mental, and neurological health, vegetarians have to reduce the N6 to N3 ratio with an additional intake of direct sources of EPA and DHA, regardless of age and gender. This comes from source number 9. Dietary issues in Hong Kong vegetarians versus omnivores. Compared with omnivores, vegetarians had higher serum concentrations of polyunsaturated and monosaturated fatty acids and lower saturated fatty acids, long-chain omega-3, and trans fatty acids. Although the Chinese vegetarian diet may be beneficial for heart health and that antioxidant and fiber intakes are higher and saturated fats lower, the EPA and DHA, due to omission from dietary source and suppressed formation by high linoleic acid level, the presence of TFA in the diet may exert an opposite effect. Our conclusion: there are some favorable features in the serum fatty acid profile in the Hong Kong Chinese population with respect to cardiovascular health, but the consumption of TFA is of concern. The Chinese vegetarian diet also contains some adverse features. This is from source 10. Testosterone deficiency in LVO vegetarians diets versus mixed diets. Our results: the vegetarian diet resulted in a lower total T level compared with the mixed diet. During exercise after 6 weeks on the diets, total testosterone was also significantly lower on the vegan or LVO diet than on the mixed diet. In conclusion, 6 weeks on a lacto-ovo-vegetarian diet caused a minor decrease in total testosterone and no significant changes in physical performance in male endurance athletes compared with a 6-week on a mixed meat-rich diet. This comes from source 11. Effects of a vegetarian diet on plasma prolactin, testosterone, and DHEA in men and women. This study reports the effect of a vegetarian diet and dexamethasone administration on the hormone status of healthy Caucasian men and premenopausal women. A lower nocturnal release of prolactin and testosterone occurred in men fed a vegetarian diet, while women, dexamethasone administration decreased the normal nocturnal release of prolactin and caused a greater decrease of plasma DHEA. These results show that diet modification can induce hormonal changes. This may be beneficial for patients with breast and/or prostate cancer. This comes from source 12. Dietary weight loss of low-carb, high-fat, or protein diets versus low-fat, near-vegan diets in premenopausal women. Weight loss was greater for women in the Atkins diet group compared with the other diet groups at 12 months, and mean 12-month weight loss was significantly different between the Atkins and Zone diets. At 12 months, secondary outcomes for the Atkins group were comparable with or more favorable than the other diet groups. In this study, premenopausal overweight and obese women assigned to follow the Atkins diet, which had the lowest carbohydrate intake, lost more weight and experienced more favorable overall metabolic effects at 12 months than women assigned to follow the Zone, Ornish, or Learn diets. The Atkins group lost more weight, had greater decreases in blood pressure, greater increases in HDL cholesterol, and greater decreases in triglycerides. The Atkins dieters were about twice as likely to make it to the end of the study, indicating that the Atkins diet was easier to follow. This comes from source 13. Now that we've established a precedence of various nutritional deficiencies within the LVO and vegetarian diets, let me discuss a few myths that are purported amongst mainstream nutrition activists. Egg consumption and the risk of coronary heart disease and stroke. Conclusions of this study. Higher consumption of eggs, up to 1 per day, is not associated with an increased risk of coronary heart disease or stroke. The increased risk of coronary heart disease among diabetic patients and reduced risk of hemorrhagic stroke associated with higher egg consumption in subgroup analysis warrants further studies. This comes from source 14. Egg consumption raising HDL levels and the change from small LDL to large LDL. Our first study. LDL size was significantly smaller in CAD cases than in controls in a prospective, population-based study. These findings support other evidence of a role for small, dense LDL particles in the etiology of atherosclerosis. This comes from source 15. Second study. The objective of this study was to evaluate whether carb restriction and egg intake modulate circulating carotenoids and affect the concentrations of plasma apolipoproteins, lipoprotein size, and subfraction distribution. Only those subjects from the egg group presented higher concentrations of these two carotenoids in plasma, which were correlated with the higher concentrations of large LDL observed in the egg group. These results indicate that carb restriction favorably alters VLDL metabolism and apolipoprotein concentrations, while the components of the egg yolk favor the formation of larger LDL and HDL, leading to an increase in plasma lutein and zeaxanthin. This comes from source 16. And the third study. Dietary recommendations aimed at restricting egg consumption should not be generalized to include all individuals. We need to acknowledge that diverse, healthy populations experience no risk in developing coronary heart disease by increasing their intake of cholesterol, but, in contrast, they may have multiple beneficial effects by the inclusion of eggs in their regular diet. This comes from source 17. Saturated fat and cardiovascular disease. A reduction in dietary saturated fat has generally been thought to improve cardiovascular health. However, a meta-analysis of prospective epidemiologic studies show that there is no significant evidence for concluding that dietary saturated fat is associated with an increased risk of CHD or CVD. More data are needed to elucidate whether CVD risks are likely to be influenced by the specific nutrients used to replace saturated fat. This comes from source 18. Questioning the role of saturated and polyunsaturated fatty acids. The harmful effect of dietary SFA and the protective effect of dietary PUFA on atherosclerosis and CVD are now in question. This comes from source 27. Consumption of grains and dietary effects. Because of its high density of negatively charged phosphate groups, phytic acid, or PA, forms very stable complexes with mineral ions, rendering them unavailable for intestinal uptake. The first step in mineral absorption requires that the mineral remains in the ionic state. As the PA content of the diet increases, the intestinal absorption of zinc, iron, and calcium decreases. The inhibitory effects of PA on magnesium or copper are more controversial. Nevertheless, PA does not occur alone in foods and is often consumed with various compounds. Phytates are always present in a vegetal matrix composed of fibers, minerals, trace elements, and other phytomicronutrients. This comes from source 19. Secondly, there are studies that show non-celiac wheat sensitivity. Perhaps there is a trend that grains are not a wholly viable solution to human nutrition. From source 20, our data confirm the existence of non-celiac wheat sensitivity as a distinct clinical condition. From source 21, these findings indicate that excessive gluten intake can induce changes in the jejunal mucosal architecture in susceptible individuals who do not have overt celiac disease. Dietary protein and skeletal health. Summary. Recent epidemiological, isotopic, and meta-analysis studies suggest that dietary protein works synergistically with calcium to improve calcium retention and bone metabolism. The recommendation to intentionally restrict dietary protein to improve bone health is unwarranted and potentially even dangerous to those individuals who consume inadequate protein. This comes from source 22. High-protein weight loss diets and purported adverse effects. Conclusion of this study. It is clear that the American Heart Association Nutrition Committee's statement on dietary protein and weight reduction contains misleading and incorrect information. Certainly, such public warnings should be based on a thorough analysis of the scientific literature, not unsubstantiated fears and misrepresentations. For individuals with normal renal function, the risks are minimal and must be balanced against the real and established risk of continued obesity. Let me talk briefly about the China study. Campbell's book, The China Study, Startling Implications for Diet, Weight Loss, and Long-Term Health, hit the shelves in January 2005, and details the turning points in his postgraduate research that led him to become a famed opponent of animal foods and an advocate of the vegan diet. It takes the reader on a tour through Campbell's early animal experiments, which he interpreted to implicate animal protein as a primary cause of cancer, through the massive epidemiological study after which the book was named. Only 39 of 350 pages are actually devoted to the China study. The bold statement on page 132 that eating foods that contain any cholesterol above 0 milligrams is unhealthy is drawn from a broad and highly selective pool of research. Yet chapter after chapter reveals a heavy bias and selectivity with which Campbell conducted, interpreted, and presents his research. This quote comes from source 23. I've included another in-depth analysis of this book and of the China study in and of itself, which you will find in source 24. Red and processed meat consumption and risk. Conclusions of this study. Consumption of processed meats, but not red meats, is associated with higher incidence of CHD and diabetes mellitus. These results highlight the need for better understanding of potential mechanisms of effects and for particular focus on processed meats for dietary and policy recommendations. This comes from source 25. An addendum, processed meat consumption and mortality. The conclusions of this study. The results of our analysis support a moderate positive association between processed meat consumption and mortality, in particular due to cardiovascular diseases, but also to cancer. This comes from source 26. In conclusion of this presentation, it would appear that the LVO/vegetarian diets are sustainable only from adulthood into midlife with supplementation, without an increased risk of health hazards. From prenatal development into adulthood, there is evidence to suggest growth deficiencies and nutritional deficits. After the onset of old age, the health risks become more severe with the LOV/veg diets, especially in the field of muscular health and regulation, and perhaps even cognition. It would also appear that women in particular may suffer more from the adverse effects of these diets than men. I will continue my research as time goes on, and I'd like to thank you all for listening. The complete list of my sources will be in the description below, and if anybody wants, I can publish the full text of this script that I've been reading from. It's very unusual for me to read from a script, but unfortunately this presentation is just way too long. It's about 8 or 9 pages long, 3,700 words, and I just figured I'd write it down instead of trying to remember it off the top of my head.